Yes, Junati.
Good morning to all. Hello to everyone joining today's webinar. I, Junaid Tantre, and as a co-convener of this event, I am delighted to welcome a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I'm confident that today's session will uphold its tradition of excellence, providing valuable insights from our respected speakers. So let's seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us. We are gathered today to commemorate the 79th SPSR International Webinar on Gene Polymorphisms associated with treatment outcomes of morphine among cancer pain patients, a distinguished event hosted by Society of Pharmaceutical Sciences and Research in association with Malaysian Pharmacists Society under the esteemed guidance of Dr. Shashi Alok Sir, National President of SPSR. The Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission, we are committed to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It's a great privilege to introduce Dr. Hui Yin, Pharmacy Lecturer, Department of Pharmaceutical Life Sciences, Faculty of Pharmacy, Universiti Malaya, Kuala Lumpur, Malaysia, as a distinguished speaker. Dr. Hui Yin expertise greatly contributes to our event, providing substantial knowledge and insight. Our participation is certain to enrich our discussions and expand exploration of gene polymorphism. We are grateful for her engagement and anticipate an enlightening session. It's a privilege to introduce Mrs. Monika Sabharwal, ma'am, as a convener for today's webinar, who also serves as a founder and CEO, SPSR Periodicals, and National Secretary of SPSR. Her significant involvement and leadership in pharmaceutical sciences and research communities are indicative for her commitment to the field. We are grateful to have her guidance in today's session also. I am pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished speaker, Dr. Hui Yin. Over to you, ma'am.
Thank you so much, Mr. Dinetantre. Hello, everyone. I am Monika Sabharwal, the convener of 79th SPSR International Webinar. And it is my great pleasure to welcome you all to this exciting session on the topic gene polymorphisms associated with the treatment outcomes of morphine among cancer pain patients. This webinar is organized by Society of Pharmaceutical Sciences and Research SPSR and in association with Malaysia Pharmacists Society. And we are honored to have with us today Dr. Hui Yin Yow, a distinguished pharmacy lecturer from the Department of Pharmaceutical Life Sciences, Faculty of Pharmacy, Universiti Malaya, Kuala Lumpur, Malaysia. As our esteemed speaker, Dr. Wow brings with her wealth of expertise in the field of pharmacogenomics and will undoubtedly provide valuable insights into this crucial area. This webinar has garnered significant interest with over 1,500 registrations from more than 22 countries. This overwhelming response underscores the global significance of this particular topic and the growing interest in advancing our understanding of pharmacogenomics and its clinical applications. As we celebrate the 79th webinar in this SPSR series, I would like to express my heartfelt gratitude to all of you for your support. Over the course of this journey, we have received a tremendous encouragement and a positive feedback, which serves as a constant source of motivation for our team. It reaffirms our commitment to uplift the pharmacy profession and creating a platform for the meaningful knowledge exchange. At SPSR, we continuously strive to organize webinars on the wide range of the topics, often inspired by the suggestions from our audience. Your active participation and interest drive us to bring you a thought-provoking sessions featuring renowned speakers from around the globe. I am also excited to announce that the SPSR Life membership will open soon. Joining SPSR offers you an opportunity to become a part of vibrant community dedicated to advance the pharmaceutical sciences. The membership provides access to the invaluable resources, professional growth opportunities, and the supportive network of experts and peers who are passionate about the pharmacy and the healthcare. For more detail, please visit our official SPSR website. Furthermore, we invite national and international institutions to collaborate with us through the Memorandum of Understanding MOUs for the future activities. Please visit our official SPSR website for more information and access to the proposal form for the collaborative ventures. Lastly, we encourage you to share your suggestions for the future topics and the speakers. Your inputs help us to create webinars that address the interest and the needs of our audience. We sincerely thank the SPSR team for relentless efforts in organizing such webinars. I also thank our audience for attending it regularly, as your participation and suggestions inspire us to work harder and continue delivering the impactful sessions every time. Before we commence, I would like to warmly welcome Dr. Shashi Alok, our National President, whose visionary leadership has been crucial in the impressive transformation of the SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Mr. Junaid Tantre, the co-convener of this webinar, and the organizing committee members, Dr. Pradeep Gulani, Dr. Sanjay Nagdev, Dr. Saurabh Kose, Mr. Ankur Agarwal, Mrs. Nidhi Mittal, and Mr. Rishabh Banot. Their tireless efforts have played a very crucial role in transforming of the vision of this event into a reality. Join the SPSR Vibrant Community, or over 40,000 pharma professionals actively engaging across the LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, grow with the top-tier webinars and very exclusive content. Do not miss out this opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSR's YouTube channel and enabling the notification bell icon for the instant alerts. Follow our journey in advancing the pharmaceutical sciences that will benefit the society. Let us now look forward for today's insightful session. I am confident that Dr. Hui Yin Ma'am talk will be rich in experience that will benefit all of us. Thank you for being with us, and I hope you will find today's webinar both informative and inspiring. Over to you, Mr. Dunay Tantre.
Thank you so much, ma'am. I really appreciate for granting me the opportunity to present the webinar. And now we will start with a short introduction of guest profile. Dr. Yao Hu Yin, ma'am, is a lecturer in pharmacy at Faculty of Pharmacy, Universiti Malaya, with a strong foundation in both academia and professional practice. She earned her bachelor's and PhD degrees in pharmacy from University Kebangsaan, Malaysia in 2009 and 2012 respectively, and is a registered pharmacist with the Pharmacy Board Malaysia. Dr. Yao's research interests are multifaceted and encompass several key areas. Her primary focus lies in neuropharmacology, where she investigates the complex interplay between molecular mechanisms and drug responses in neurological disorders, particularly epilepsy. Her research aims to bridge the gap between animal and human studies by employing advanced pharmacological approach to gain a deeper understanding of disease processes and identify more effective therapeutic interventions. Furthermore, Dr. Yao is actively involved in research related to pharmacogenomics and precision medicine. She investigates the genetic factors that influence individual drug responses with the goal of developing personalized treatment plans that maximize efficacy and minimize adverse effects. In addition to her research in basic and translational sciences, Dr. Yao conducts research in pharmacy practice also. This area of our work involves utilizing both qualitative and quantitative research methodologies to study various aspects of patient care, including medication use and management, drug-related problems, patient monitoring, and quality of life. These studies aim to identify and address critical issues in pharmacy practice and ultimately enhance patient outcomes. Dr. Yao's research employs a diverse range of methodologies, include omics technologies such as transcriptomics, genomics, and metabolomics, as well as advanced pharmacological techniques. She has successfully secured several research grants to support her ongoing investigation. Dr. Yao's contribution to the field of pharmacy are significant, and her research has the potential to make a substantial impact on the development of new therapies and improvement of patient care. It is with a great satisfaction and pride that we declare that we have exceeded 1,500 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from nations include Asia, Azerbaijan, Bhutan, Canada, Egypt, Indonesia, Iraq, Kenya, Malaysia, Morocco, Nepal, Nigeria, North Macedonia, Oman, Pakistan, Philippines, Saudi Arabia, Sudan, UAE, USA, and Vietnam. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each and every person who has taken a moment to participate in the significant event. Thank you for all joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Yao Hu Yin, with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Thank you, Dr. Junaid, and also Mrs. Monika for your kind introductions. Without further ado, I will share my screen. Just give me a few minutes. Just for the screen check.
Yes,
Is it
ma'am.
visible?
It's visible.
All right. Thank you. All right, without further ado, I would like to take these opportunities to thank Society of Pharmaceutical Sciences and Research, as well as Malaysian Pharmacists Society for the opportunities to organize these webinars. So I would like to share on my work on gene polymorphisms that are associated with the treatment outcomes of morphines among cancer pain patients. So allow me to share a couple of minutes to talk about my country. I'm coming from Malaysia. So if you look at the locations of Malaysia, it's located in the Southeast Asia and is bordered by the Thailand as well as Singapore and also Indonesia. So yeah, the capitals of Malaysia is Kuala Lumpur. So I'm from Kuala Lumpur. Right, we do invite all of you to visit us when there is the opportunities. Right, so let's look into my university. From University of Malaya. Or in Malay, we call it as Universiti Malaya, and I'm from Faculty of Pharmacy. So previously, we was known as Departments of Pharmacy under the Faculty of Medicine since 1995, and the last 6 years, we successfully upgraded to Faculty of Pharmacy, which is fully operated since 1st September 2019. Right? Right, so let's look at the topics today. I think cancer pain is not something new for everyone because cancer pain is actually very prevalent and it's one of the most common symptoms that occurs among cancer pain patients. However, we always link these cancer pains with prevalence care. But in fact, cancer pains can come from the tumors or the treatments, and it can also continue after the successful cancer treatments end. So basically, the pain can actually occur throughout the process, during the cancer's developments, during treatments, or even after the treatments. Therefore, controlling pain is actually an important part of the cancer pain treatments. So if you look at the cancer pains and its prevalence, so 55% of the pains actually happens during anti-cancer treatments, and 39% actually happens after the curative treatments, and 66%, which is the highest prevalence amongst those with advanced metastatic and also terminal disease patients. So from this, we actually know that how prevalent cancer pain is among cancer patients. And unfortunately, it's estimated that more than 30% of the patients worldwide receive inadequate pain relief. And from the studies, it shows that suboptimal management of the cancer pains is actually the major source of stress for cancer pain patients. And this can negatively impact on their quality of life. So the main goal of pain management is to achieve levels for accepted quality of life. So we want to control the pains so that everyone actually at acceptable quality of life levels. So the cancer pain itself is actually involve very, very complex events. So in terms of the underlying mechanisms, is still under investigations. We can actually see that a few mechanisms being proposed, but it's still not, the exact mechanism is still unknown, right? So the type of cancer pain classifications actually help us to sort of understand the mechanism of cancer pains. So if you look at the type of cancer pain, it can divide into 3 classifications: nociceptive pain, neuropathic pain, or a mix of both. So if we look at nociceptive pains, basically it's related to the pains due to the damage or injuries of the bone, the organs, as well as muscles. So it can be subdivided into somatics as well as visceral. So somatic pain usually described as sharp, aching, and throbbing pain, whereas visceral pains, which actually involve the internal organs, often described as dull and deep, as well as cramp-like pain. So for neuropathic pains, basically the mechanism is related to the damage of the nerve itself. And it can actually cause a very, very sharp pain. And this pain can be hard to control by the conventional analgesic, right? So in terms of cancer pain management, the World Health Organizations recommended the three-step analgesic ladders. I think all of you are quite familiar with this. So you have seen this before. So the first step usually involves non-opiates with or without adjuvants. And second step involves the use of opiates for mild to moderate pain with or without non-opiates and adjuvants. And the third step involves opiates for moderate to severe pain with or without non-opiates or adjuvants. So if we look at these three-step analgesic ladders, we can actually see that a comprehensive pain assessment is required before the pain management started. And also ongoing monitoring of pain is required in order to adjust dose accordingly. And the most commonly used adjuvants for cancer pains include steroids, antidepressants, and also anticonvulsants. So based on the Ministry of Health Malaysia, is actually adapted the guidelines from the three-step analgesic ladders from WHO and added with the pain score. So we can see step 1 here, we have pain score 1 to 4, which actually indicate mild pain. Step 2, pain score of 5 to 6, moderate pain. Step 3, pain score of 7 to 10, which actually indicates severe pain. So for non-opiates, the recommended drugs are paracetamol, NSAIDs or COX-2 inhibitors with or without adjuvant, which is similar with the step 1 of the WHO three-step analgesic ladder. And step 2 involves the use of weak opiates, such as tramadol, dihydrocodeine, as well as codeine. And the third step involves strong opiates, which actually include morphine, fentanyl, oxycodones, and also methadones. So from the literatures, we can see that opiates have been used widely, and it remains as drug of choice for the treatments of moderate and severe cancer pain. So if we look at morphines, it has been used for many years and it still remains as the first-line strong opiates, which is recommended by the WHO guidelines because of its availabilities and also costs. So, however, up to 30% of the patients do not respond to morphines and have poor treatment outcomes. So these 30% might not getting beneficial effects from morphines, means that the pain is not controlled, or they actually encounter some unwanted side effects, particularly hallucinations, drowsiness, severe constipations, nausea as well, right? These inter-individual differences have been shown to related to inter-individual genetic variations. So means that genetic variations play some roles to or play some it's actually a cause for these inter-individual differences. So before we look at the inter-individual differences, I will share some mechanism of morphines in terms of its metabolism and as well as its mechanism of action, so that we can actually relate it more with the genes later on. Okay. So if you look at morphines metabolisms, so morphines will be transported into the liver cell through the influx transporters. And here in the liver cell, it will be metabolized by the UGT enzyme, which is the uridine diphosphate gluconestyl transferase enzymes. And these enzymes are mainly UGT2B7 or UGT1A1. And after metabolisms, it will actually produce two main metabolites, which is morphine 3 gluconides and also morphine 6 gluconides. So the active form is actually the morphine 6 gluconides. So after the metabolisms, these metabolites will be transported out from the cell through the efflux transporters. You can see here MRP2. So MRP is actually the multi-drugs resistance proteins, or also commonly identified as ATP transporters, which is ATP binding cassettes transporters. Right. So in terms of the mechanisms, so we know that the active metabolites is morphine 6 gluconides and M6G, and also the morphine, the parent drug itself, they are active and works towards opioid receptors, which is mu1. And there are 4 agonisms. So basically, they work on the mu1 receptors and provide the pain-killing effects. And in terms of the other receptors, morphine and M6G, they also bind to mu2 receptors, which actually believed to have to cause a lot of adverse effects, such as respiratory depressions, constipations, and sedation as well. So from what we can see now, one challenge with morphine use is due to inadequate pain relief. So there is a lot of concerns on the addiction's effects from the morphine, and as well as the adverse effects that we mentioned earlier. And the third factors is due to the inter-individuals differences. So there are 30%, at least 30% of the patients are not responding to the morphines, or they are experiencing adverse effects, drug toxics, after the morphine use. So one way to explain the inter-individuals differencing is through pharmacogenomics. So basically, pharmacogenomics actually provides an understanding how these genetic variations can affect the treatment's outcome. So for our studies, we're focusing on morphines. So based on the studies, genetic variations have been hypothesized to account for approximately 20 to 95% of the inter-individuals variations in drug response. And the pharmacogenomic studies provides an effective, safe therapies individualized for each patient. So in our studies, we use this pharmacogenomics approach to understand why these inter-individuals variations actually occurs in our populations. Right? So if you look at the genetic variations, the single nucleotide polymorphism, or we call it SNPs, is actually one of the most common genetic variations, and is actually related to a single nucleotide base that being replaced by another nucleotides in the DNA. So if you look at here, so we have the G here, for example, guanides, and being replaced by the T here, thymines. So this actually is one of the examples of a single nucleotide polymorphism, and these mutations actually cause to different alleles that happens in our populations. And the locations of the SNPs can be in the protein coding sequence, regulatory regions, as well as exons entrance of the genes. So if we look at what have been done for the pharmacogenomics in cancer pain, basically most studies are actually focusing on Caucasian populations, and there are some significant results have been reported. For example, OPRM1, which is the opioid receptors, mu1s. So as we learned just now, opioid receptors, mu1 is actually one of the primary targets for morphines. So it's actually the important receptors where the morphines will actually work on and shows the effects. So in terms of the OPRM1A118G variants, it actually show a difference in significant difference in terms of the opioid requirements and pain response. Whereas ABCB1, so it's actually an importance of efflux transporters that will actually transport the metabolites of morphines out of the cell. So this actually affects the viabilities of the drugs in the cell, and it also might contribute to the side effects. So side effect, so the studies actually show that ABCB1's C3435T variants associated with central side effects such as drowsiness and hallucinations. Whereas COMT variants, so if you look at COMT, it's slightly different from other enzymes which actually involved in metabolism. And this COMT is actually involved in pain modulations. So it actually works through a dopamine-mediated pathway. So with those people that with COMTs impairment, enzyme impairments, they actually shows to have increased dopamine concentrations, and these dopamine concentrations can actually suppress the productions of endogenous opioids, and this leads to the upregulations of opioid receptors. So this explains why COMT variants actually affects the morphine efficacy and also central side effects. So, as I mentioned just now, most of the studies have been done in Caucasians in European country, and they're lacking of informations or conclusions coming from Asian populations. The associations of the genetic variants with the cancer pains treatment outcome from the opioids in Asian populations actually remains unclear. So this is why we actually did a scoping review on these topics in order to determine the sleeps associated with the treatment's response amongst patients that using opioids in Asian populations. So from our review, we actually identified our articles from the MBase as well as midlines. We were able to get around 658 papers. So from these 658, we removed the duplicates or those articles that published before 2000, year 2000. And afterwards, we did a screening on the title and abstract, and followed by full text review. And based on our inclusion and exclusion criteria, we actually included 14 papers in this review. So just to share some of the results from our scoping review. So these are the list of the genes that reported with significant association in Asia populations. And these genes lists are not particularly for morphine only. It also for others opioids, for example, like tramadols, fentanyl, oxycodone. So this is some things that we have summarized based on the significance result. So particularly, I want to highlight on morphines. So there are few genes that related to morphines. So UGT2B7, this is actually one of the genes that related to the metabolism of morphine just now. So morphine actually metabolized through a conjugation process. So UGT2B7 is actually one of the main enzymes that work on the morphine metabolism. Another gene is ABCB1. So ABCB1 is actually an efflux transporters. So it also shows significantly affects the treatment outcome. And then the next one is OPRM1. So this is the main targets for the morphines to work on the receptors. And then the next one is COMT, which actually related to the pain modulation pathway. So these four genes are mainly related to morphines. Right. Next, I would like to share some details on the studies that work on the polymorphism in terms of the opioids pharmacokinetics. So we actually found two papers from China and also Japan's actually work on the UGT2B7 and also CMT variants. And these two studies actually found that the plasma concentrations of the morphines are associated with the these genes variants, right? And then the next parameters, they look into opioids dose requirements. So there are three papers here. One is from Lebanese, another one is actually working on Japanese. Another tools are working on Japanese. So basically they notice that OPRM1 variants and also COMT variants have a significant effects in terms of the opioid dose requirements. And then the next one is about the polymorphism that's significantly associated with pain control. So we can actually see that another studies from China actually work on the UGT2B7, and it actually show associations of the genes polymorphisms with the pain control. So from our studies, from our reviews, we actually noticed that the some of the SNPs that needs further explorations, particularly for Asian populations. So we can actually see that mainly mainly Japanese and also Chinese populations have been explored. Others part of the Asian populations are still remains unclear for in terms of the associations parts. And even though some SNPs actually show significance, but there are insufficient evidence. So maybe due to the controversies of the results. So this is actually telling us that more studies have to be done in the large scale studies, so that we can actually explore in terms of the associations of these SNPs with the treatment outcome of morphines or opioids. So some highlights from our scoping review. So we actually noticed that some SNPs are relatively important in Asia populations. For example, OPRM1, A11G. This genetic variance is actually more frequent in Asians with 40 to 60% compared to Europeans, which is around 15%. So this actually indicates that some SNPs are relatively important in Asian populations compared to others, for example, Caucasian populations. And there's some contradictory results. So for example, COMTG4728, the AA genotype actually shows low morphine concentrations and required low doses of morphine among Japanese. But in a Caucasian studies actually shows that A-alleles reduce analgesic efficacy. So basically the results are contradictory. And as I mentioned just now, most studies are actually focusing on the patients mainly from the East Asia ancestry, which is Japanese and Chinese. So we can't actually conclude for those from other parts of Asia, for example, Southeast Asia or the central parts of Asia, as well as Southern Asia. So there are a lot of research gaps that need to be addressed from here. And another thing that we notice is because every study is actually using different outcome measures. So some of them use opioid dose requirements, some of them using durations of treatments as their outcome measurements, and some of them assess the pain severities. And different tools was used as well. So in terms of that, it actually limits the abilities to compare the findings between the study. So from our scoping review, it gives us some clues in terms of how genetic factors actually affected Asian populations. And here come our research questions. What are the genetic variants that present among cancer patients in Malaysia populations? So for your information, Malaysia is actually a multi-ethnicities country. So we are actually comprises of mainly Malay, Chinese, and India ethnicities, but also some of the indigenous ethnicity as well. So we don't know whether there are genetic variants that presents among cancer patients in our populations. And the second question is, are there any correlations between genetic factors and the pharmacodynamics of the morphines? So particularly for pain control, dose requirement, as well as side effects among our cancer pain patients. So this is why we conducted studies in Malaysia. I call it primary studies because the sample size is slightly small compared to the one that we wish to get earlier. So we obtained an human ethics clearance before patients' recruitments. And our patients were recruited from National Cancer Institutes between July 2020 until July 2022. You might wonder why we take 2 years for patients' recruitments. So this is mainly due to the COVID-19's restrictions. So we have the restrictions to access to the hospitals, and some of the patients are not following up during the COVID time. So this actually giving us difficulties in terms of patients' recruitments. And we're only able to recruit 130 cancer patients. And from these 130 patients, we have 66 patients were treated with morphine, which is around 50.8%, based on our inclusions and exclusions criteria. And in terms of the data collections, we actually assess the pain severity based on a validated assessment tool, which is the brief pain inventory, or in short, we call it as BPI. I will explain more about BPI later on. And also we developed a questionnaire to actually determine the incidence and severities of the adverse effects that experienced by the patients. And we also retrieve others medical or clinical information through our medical databases. And from our recruitments, we're able to actually get 57.6% of our morphine patients to complete the questionnaire for the pain severities as well as adverse effects. So this is how brief pain inventories assessment tools looks like. So it's slightly different from the VAS, which is a visual analog scale. So this brief pain inventory is actually asking some more in-depth questions related to pain locations, as well as the pain severities at different time points. For example, in the last 24 hours and the pain right now. And also assess the effects of the pain on patients' daily activities. So for the DNA parts, for the genotypings, we actually collected patient saliva samples using Origin's saliva collection kits. So initially, we actually wanted to collect patients' blood samples, which is more direct and fastest way for us to extract our DNA. But there's a limitations for cancer patients because most patients are actually undergoing chemotherapies and having neutropenias. Because of neutropenias, we couldn't actually extract a lot of DNA from the white blood cell because the white blood count is low. And we only can get the DNA from the nucleus, which is available in the white blood cell, and the red blood cell has no nucleus. So this is one reason we actually change our methods from blood sample collections to saliva sample collections, which is less invasive and also overcome the issues of neutropenias among cancer pain patients. However, there are a few challenges for us. Due to the saliva samples collections, our patients mainly actually having dry mouth conditions due to the morphine treatments. So it's actually a bit hard for us to get the saliva samples. And besides that, there are a lot of contaminants in the saliva samples. So this actually requires more optimization step in order for us to extract the DNA. But we're quite lucky. We are able to extract all the DNA based on the protocol, as well as the qualities are acceptable. So after the DNA extractions, we actually proceed with the genotyping. We use Tagman genotyping assay. And afterwards, we confirm our result using a single sequencing methods just to validate whether our result is accurate. So we actually selected around 10 to 15 samples and sent for single sequencing. So these are actually the demographic characteristic of our patients. So we can see here our patients, the age is around 52 years old, and mainly are females. And in terms of the ethnicities, it's actually representative of our Malaysia's populations, which mainly are Malay, followed by Chinese and India. And we also look into patients' cancer pain stage. So most of our patients are stage 4 patients. And in terms of the ECOG score, which is one of the scorings that used to determine the patient's functioning levels, it actually consists of 6-point scale. So 6 is actually, it's actually starting from 0 to 5, and 5 is actually indicating death, right? So in terms of the alleles and genotyping frequency of the SNPs, so all the alleles and genotypes frequency will in Hardy-Weinberg equilibriums, which means that the distributions of the genes are actually past the QC quality check, and they are actually without genotyping errors, right? And from the mutants alleles frequency, we can actually see that ABCB1s, the mutants frequency is actually the highest. The prevalence of mutant alleles is highest amongst compared to the rest. And OPRM1 is actually the second one. And if you look at the mutant allele frequency here, we compare with others Asian populations, it's actually aligns with others Asian populations. So except for the COMT. So COMT's variance is relatively low prevalence in our country, and which actually indicates that the genetic polymorphisms in COMT might be less likely to affect our patients' treatment response because of the low prevalence. So in terms of the analgesic regimens and the concurrent medications of patients that receive morphines, so most of patients are actually taking oral solutions, 75%, followed by oral tablets, 41%, and injections, 14.8%. And the total fixed morphine oral dose is actually around 34 milligrams. So if we compare the patient's cancer stage just now was around stage 4, mainly of stage 4 patients. But in terms of the fixed morphine dosage is slightly lower. So this one of the explanation is in these studies, we didn't actually take into the consideration, we didn't actually take into the account for the PRN, the basis, the pain dose, the dose that requires for breakthrough pain. The dosage that requires for breakthrough pain is not actually counted in our studies because we couldn't actually retrieve this data from the medical records. So this actually explains why our total fixed morphine dose is relatively lower, even though most of our patients are actually coming from palliative care background. So in terms of morphine regimens, so most patients are actually having combined opiates. So morphines with others like methadones, we have 4 of them. There are fentanyls and tramadols, and also some of them are actually combined with adjuvants like anticonvulsants and also antidepressants. And other things to highlight, most of the patients are actually receiving laxative as a preventive measures for constipation side effects from morphines. And also some of them actually, 57% of them actually receiving antiemetics. So if you look at the associations of these genetic polymorphisms and the morphine dose requirements, we notice that there's not significantly found for this genetic variants, even those other studies shows that ABCB1s and COMT variants might require higher doses of morphines. So in terms of the pain severities, so we actually use the BPI assessment tools. So if you look at the pain score, it was in the last 24 hours. Basically, the score is around 5.5, a median of 5.5, which actually indicating that most patients are actually having moderate pains, because the pain score of 5 to 6 actually indicates moderate pain. But the pain score for right now is actually 1.5. And we can actually see there's variations in terms of pain control, particularly for the last 24 hours. It's actually telling us that maybe morphine dose is inadequate in our populations. Besides that, we actually did association studies for the pain severities, and we also noticed that pain severities was not significantly associated with genetic factors. And then we further look into the incidence and also severities of the morphine-related adverse effects. So if we look at here, drowsiness and constipation is actually the highest incidence. So even though most of our patients already treated with laxative, more than 80% of our patients receive laxative as preventions, but there are still 27 of them, which is 71% actually having constipations. And there are others reported side effects like serotrauma, which is dry mouth, and also fatigues. So in terms of the associations of the genetic variants and the morphine-related adverse effects, we do identify ABCB1's variants actually significantly associated with some of the adverse effects like pruritus, dyspneas, headache, as well as urinary retentions. And for dyspneas is actually reported by previous studies as well. That happens amongst those children that treated with opioids. And for pruritus and headache, as well as urinary retentions, our studies is actually the first reports that showing these associations. Another significant result is related to the COMT variants with the occurrence of headaches with those COMTs RS4646312C alleles. So this was not reported previously. So another genetics variants that significantly associated with the morphine treatment outcome is actually dyspnea. So dyspnea is actually showing to be significance with OPRM with OPR and RS1799971G allele. So these studies were supported with the previous studies that done in mice, and where the GG genotypes are actually showing exhibitions of asthma-related features. However, contradictory results be reported amongst Caucasian patients, where the wild type of A allele carriers was significantly associated with respiratory depressions. So the result is actually the other way around. So I'd like to discuss some of the study limitations that related to our research. So the first limitations is due to the COVID-19 restrictions that actually impacted our timeline for patients' recruitments, where we took 2 years to complete the patient recruitments as well as data collections. And because of this, we have a very small sample size. So these actually reduce the statistical powers of our studies. And one of the reasons that we have low response for our questionnaires, because that we actually contacted our patients through phone call because of the COVID-19 restrictions. So we actually contacted our patients and asked them to complete our questionnaire terms of the pain severities through BPI assessment too, and also the adverse effects questionnaire. And the response rates were around 57%. So one of the reasons of this low response rates is because a lot of people are actually worried about scammer call. So they treat us as scammer as well. So they rejected our call. That's why we couldn't actually contact or get through our patients, even those we already obtains their consents earlier. And the last limitations is because it's related to the morphine side effects. All these side effects are self-reported through questionnaire, and it was not confirmed by doctors. So because some of the side effects could be quite could be hard to differentiate between the cancer itself and also the drugs side effects. So this actually might be our study's limitations. However, the findings from our studies actually could contribute to the current research on the role of pharmacogenomics in terms of the treatments of cancer pains in Asia's patients. So from our studies, we actually shows that there are some significant associations, particularly for the morphine-related adverse effects with genetic variants. So these SNPs might be a potential pharmacogenomics biomarkers in tailoring cancer pain treatments. So in terms of the future studies, so our study is only focusing on certain genetic variants, and we actually work through using a genotyping approach. So basically, we didn't actually see a full pictures or combinations effects of several SNPs together. So if we can have a halotype-based analysis, which actually combine the effects of several SNPs together with the high linkage disequilibriums, it actually helps us to detect or help us to improve the accuracy of our results. And besides the halotype-based analysis, another one is actually the GWAS, which is genome-wide association studies. It actually screened the entire genomes. So these methods or this approach can actually help us to identify the genetic factors that involve in pain and also morphine response in Asian populations. So as conclusions from our studies, it shows that even though the morphine dose requirements and pain severities were not associated with the SNPs that we studied in our research, but the result actually telling us that there's significant association with between the genetic variants and also the morphines related adverse effects. So some of the findings are firstly reports in our studies. So this finding should be further validated in futures genetics association study in a larger cohort, so that we can actually confirm the role of these potential biomarkers in the treatments of cancer pains in Asian populations. So with this, I would like to thank my master student, Shoba Elizabeth, who actually completed the studies, particularly during the COVID-19's influence, but she managed to actually go through all the obstacles between the studies durations. And I would like to thank our funding sponsor, which is the Ministry of Higher Education and the Fundamentals Research Grant Scheme, Malaysia. And would like to thank my collaborator as well, Dr. To from Taylor University, Dr. Vijaya Prakash from UniSA, Australia, and Dr. Muhammad, Dr. Chua from UK and Malaysia, and Dr. Nusaiba from Universiti Malaya, and Dr. Hasriza, as well as Dr. Fauzia from National Cancer Institutes, Malaysia. Right. With this, I would like to end my presentations. Thank you.
Thank you so much, ma'am. It was such a brainstorming and exceptional lecture. Thank you so much, Dr. Yao, for delivering such a fascinating and insightful lecture on gene polymorphism associated with treatment outcomes of morphine among cancer pain patients. Your in-depth exploration of the genetic factors influencing morphine efficacy and safety has provided us with a valuable knowledge. I think that bridges pharmacogenetics and personalized medicine. The clarity and detail of your presentation have truly enriched our understanding of this complex and critical area. We deeply appreciate your time and effort in sharing your expertise with us today, and we are grateful for your remarkable contribution to our 79th SPSR International Webinar. And thank you so much, ma'am. Before we could conclude, we have received your questions, which I'll ask one by one. The first question we have received is, why do genetic polymorphism result in such a wide inter-individual variability in morphine response?
Thank you, Dr. Junaid. This is a good question. Basically, how genetic polymorphism can influence individual response to the drugs is related to the genes, the genetic variations, because the genetic variation itself actually affects both pharmacokinetics and also pharmacodynamics of the drugs. For example, the genetics variations that I mentioned just now, ABCB1, which is the one that can change the activities of the transporters. So it can affect how the drugs being effluxed out. So all these are actually related to the genetic polymorphism, and it can affect the individual's response towards the drugs. Hope I answers
Thank you, ma'am.
questions.
We have, yeah, of course, of course. Thank you so much. I hope the person has got an answer. Now, second question will be, can your findings on ABCB1, COMT, RS4880 polymorphisms be used to inform clinical guidelines for morphine prescription?
Yeah, well, this is actually a very practical question. So even though the findings from our studies actually suggest the potentials of these NIPs to be used as biomarkers in personalized medicines, but basically the whole thing depends on the strength of the evidence and also the consistency of the findings. Because like a lot of significant results actually first reported in our studies. So basically we need more replications of the result to confirm. Particularly our studies actually involve small sample size. So we need a more large scale and well-designed studies to further confirm of these findings in order for us to establish the clinical utilities.
Great.
So for now, it's actually not ready for clinical guidelines. Yeah.
Thank you so much, ma'am, for this one. So we have one more. What steps can be taken to overcome the challenge associated with the small sample size in pharmacogenomic studies? Yes, I think also one of my query also now.
Yeah. I believe sample size is actually one of the key determinants in a lot of studies. So we look at sample size, it can affect the results of the studies, and it also affects the statistical power of the whole studies. So to overcome this challenge, what I can actually propose is actually to collaborate with others researchers. So this is a studies that we have done in Malaysia. Perhaps we can actually extend to others populations, and then we can actually pull the whole data together. And this is how we can actually collaborate to increase the sample size and enhance the statistical power. Yeah.
Exactly.
So with this, I actually would like to encourage those who want to collaborate. You can actually contact me later.
Yeah, sure, ma'am. Of course. So, ma'am, we have last, then I will have my personal question at the end. And the last question will be, what are the next steps of future directions for research in pharmacogenomics to improve cancer pain management outcomes? What are the next step of future directions?
Okay. Yeah. Right. so we can actually see nowadays people talk about personalized medicines. We talk about omics, how this pharmacogenomics actually improve managements. So same for the cancer pain management, the future direction is towards personalized medicines. If we are able to identify some predictive biomarkers that can predict our patient response to a specific pain medications, basically this allows more precise and effective treatments. And with the individual's genetic profile, it also helps us to tailor the treatment plan according to our patients in order to optimize the pain control and also the minimizing the side effects.
Thank you with this one, ma'am. Now my question is, people are watching, I think from globally people are watching. So there are many budding researchers who want to, research in some area, but they are confused that how to start research, how to focus, what are the initial steps one can keep in mind to initiate in some research, or you can say a review article or anything related to this scientific community. So what is the thing that one should keep in mind, or what are the any specific steps?
Yeah.
As we have
Yeah, research possibilities are a lot, are everywhere. Basically, there are a lot of possibilities, but the most important thing is to identify the research gap, like initial literature review searchings. For example, we did a scoping reviews to identify which genetic variants that we want to focus on. Yeah, there are a lot of approach that we can adopt and we can learn from others research group. So one of it is actually systematic reviews, scoping reviews. All this actually help us to understand the areas better. So once we have these, we can actually design our studies to actually further address the research gap that happens, and this can actually increase the applications of our study results. Yeah, I hope.
thank you.
Yeah.
Of course, people will get because it will be available now, so people can see it and they can take inspiration. And I personally have gone through on your profiles, and it's commendable, ma'am. Thank you so much. So before concluding 79th SPSR International Webinar on Gene Polymorphisms associated with treatment outcomes of morphine among cancer pain patients, we extend our deepest gratitude to Dr. Yow for delivering an exceptionally insightful and thought-provoking session. Dr. Yow's comprehensive discussion on the impact of genetic polymorphisms, such as those of OPRM1, ABCB1, and CMT genes, has shed light on complexities and inter-individual variability in morphine response and adverse effects also. Our findings emphasize the importance of pharmacogenomics in advancing personalized medicine, particularly in optimizing cancer patient management for improved patient outcomes. The session has underscored the potential of genetic biomarkers to transform the prescription practices of opioids like morphine, ensuring a better efficacy and reduced side effects. It also highlighted the need for continued research, collaborative research in particular, especially in underrepresented population, to build robust clinical guidelines informed by pharmacogenomic evidence. We sincerely thank you to all the participants around the globe for their active engagement and thought-provoking questions, which made this webinar both interactive and enriching. We look forward to your participation in future SPSR webinars as we continue to explore cutting-edge topics in pharmaceutical sector. We are deeply thankful to Dr. Shashi Alok sir, the National President of our SPS Society of Pharmaceutical Research, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks to your significant inputs also. I'd also like to express our sincere appreciation to our convener, Mrs. Monika Sabharwal, ma'am, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative effort, which has broadened our understanding and fostering cooperative learning. We look forward to your participation in future SPSR webinars as we continue to explore groundbreaking topics and foster knowledge sharing in pharmaceutical sciences. Thank you once again, and we wish you all the success in the endeavors. Our upcoming 80th SPSR International Webinar on Artificial Intelligence in Pharmaceutical Care. Esteemed speaker of this event is Professor Dr. Mohammed bin Mahmood Bakri, Dean and Professor, Faculty of Pharmacy, University Kebangsaan, Malaysia. The webinar link is given in the description box. Have a wonderful day ahead. Thank you so much, ma'am. Once again, it was a pleasure, ma'am. Thank you.